spectrophotometer to measure cells. Somebody described to me the spectrophotometer and what it measures. What are the two things that the spectrophotometer can measure? Indirectly.
The total number of cells?
Yeah, it can't determine the total number of cells directly. What can it determine? It can only determine two things. It can determine how much light can pass through and how much light gets absorbed. Those are the only two things that it can determine. How much light passes through that specimen and how much gets absorbed by that specimen. And then you have to go back to those plates that you made with all those serial dilutions, and you've already recorded for each one of those tubes of serial dilutions how much light would pass through each one of those tubes. And then you can take your unknown tube and compare it to those known tubes and figure out how much bacteria is actually in that tube. Does that make sense? So you use those dilutions that you made previously, that you plated out when we were talking last time, to make essentially a standard curve. And you could plot out the absorption or the transmission for those guys. Absorption would go this way as you're going more concentrated, and transmission would go the opposite, right? Transmission is the amount of light that gets through. And then you can take your unknown, like that tube on the left there, and then you can put it into the spectrophotometer, and presuming it falls in the middle of that curve somewhere, you can determine, basically extrapolate approximately how many cells are in that unknown tube. Does that make sense? Okay. If not, come to my office and we'll draw that out a little bit and we'll talk about it a little bit more. But it's useful because remember, when you plated out those dilutions that you made, you don't know immediately how many cells are in each one of those dilutions. It takes 2 days or at least a day for them to grow out before you know how many cells were in each one of those dilutions. And so now you can use the spectrophotometer. It's very quick. In a matter of 5-10 minutes, you can get a number, an amount of absorption or amount of transmission, and you can relate it to those known dilutions that you made previously for that organism. There are other ways that we can figure out number of cells. If you think about it, you put one cell into a tube, and now it grows to 2, 4, 8, 16, 32, and so on. The more cells there are, the more biomass there's going to be. And that seems to be a term that a lot of our biology majors don't get real well. Biomass just means the mass that's been produced by the biological organisms that are living in that environment. So out in the wild, just outside of this window, we've got a lot of biomass because we've got this beautiful, looks like pecan tree growing outside the window here, and it's got lots of leaves that it just produced. Those would all contribute to the biomass. You'd take those, you'd dry the leaves out, get the water out, and what's left, that dry weight, is going to be biomass. We can do the same thing with bacterial cells. We can take those cells, put them in a centrifuge tube. We can centrifuge the cells down, pull the liquid off, and then we can take the pellet directly and weigh it. That would be a wet weight. If we really want to know the biomass, we would dry those cells, get all the water out, so that we just know how much mass is produced by the cells themselves. And so that we'd have to put into an oven to dry and be able to then measure the amount of macromolecules that those cells have produced, those cell components that they've produced. There's lots of other ways to do that. We can look at pH. We can put in a pH indicator, and you can probably conceive that one cell can produce X amount of acid. If one cell can produce X amount, then 2 cells should be able to produce 2X amount of acid, right? And 4 cells ought to be able to produce 4X amount of acid, and 8 cells 8X amount. So you can measure the amount of acid that's produced, and that will give you some rough indication of the number of cells that are there. We'll do this in the laboratory in just a few weeks. A Durham tube is a very, very small test tube. You can put one of those down inside of a larger test tube that has media in it and put it in upside down. And then when the organisms get into that small test tube inside of the big test tube, if they're producing gas, the gas will get trapped inside of that small inverted test tube, and you can measure the amount of gas. And again, X number of cells will produce X amount of gas. 2X number of cells will produce twice that much gas. And so you can determine how many cells are there. Same thing for CO2 production. That's just one way that you could do that. And then there's this ATP production. There's an enzyme called luciferase, and Luciferase is kind of cool. You already know about luciferase. You just didn't know the name yet. But what are those cute little, in the spring, they're out in the, little bugs that start flashing. Did I talk to you guys about this already? They're flashing out there, and they're just making you think, oh, that's so cute. But really, they're trying to attract their mate by flashing. And the female says, whoa, that guy's got a really cool, beautiful flasher. I want to go see what that guy looks like, and attracts the females over a little bit closer. And so scientists got curious by that ability to flash and how they did that. And they started looking at the fireflies, or what do we call it? What's the other word? Lightning bugs? Yeah. And started looking at how they do that. And they actually make this enzyme called luciferase. And luciferase can take ATP, and it can hydrolyze ATP. It's a little bit complex reaction, but essentially luciferase hydrolyzes ATP. And instead of releasing the energy from ATP as heat, it releases the energy in the form of light. So in other words, it converts the energy to photons that we can visualize. And scientists said that's pretty cool. We can use that. We can clone that enzyme, and we can put it in with a cell extract, and then we can figure out how much ATP is being produced by those cells. So you can lyse the cells, the ATP will be released, throw in a known amount of luciferase, and it'll make X amount of light depending on how much ATP is present in that extract. So it's a pretty cool method, and it's been used now for a whole bunch of things. I told my 11 o'clock class, so I'll tell you guys really quickly. America's population seems to be getting infatuated with tattoos, and tattoos are pretty interesting, pretty cool. And so I had this idea a couple years ago, and I tell every class now, because if anybody actually does it, I want to have had first conception rights on the patent so that I can get royalties off of the patent. So I've told probably 200-300 students now, I think it would be really cool if we would develop a system where you could do a tattoo, and instead of putting dyes down under the skin, you could actually put the gene that codes for luciferase into the cells down under the skin. And then once they have the gene that codes for luciferase, they'll start making that enzyme, and then instead of having a beautiful, colorful, or black grayscale, you would actually have a tattoo that when you turn off the lights, it would actually glow in the dark. They have that? Is it a chemical? Is it a chemical glow that it absorbs light and then? Okay. So, but luciferase will do it all night long, all day long. If you're in the dark all day long, because your cells are making ATP, and most of the chemical ones will fade after a few hours if they're not being recharged and absorbing the energy. So, I think that will be a multi-million-dollar seller. So I'm telling you so that when it happens, I can say, hey, I already described this to my class long ago, and just imagine having, in the daytime, you'd see nothing. And then when the lights turn off, there'd be this beautiful tattoo that starts glowing in the dark, and I think that'd be pretty awesome. So, actually, one of the students in my 11 o'clock class went and researched all that we need to do, and so we'll probably start doing that next week. It's a little bit complicated, but I think we're going to apply for the patent rights next week, because somebody else is going to think of it if we don't do that. So we've talked about a number of ways to count cells. We've talked about direct cell. We've talked about counting viable cells by plates, using that membrane filtration, using the probable number. We just talked about turbidity with that spectrophotometer. We can do biomass. We can do acid production. We can do ATP. Those are a lot of different ways that you can count cells down inside of cultures. That's the end of chapter 4. And you guys have any questions about that? I haven't looked to see how many of you viewed the videos that I'm putting on YouTube and then linking to the Facebook group, but the lectures that I'm recording, I'm putting there. The first one I looked the other day, there were a handful, maybe a couple handful of people that had actually viewed them. I'm thinking about, if I don't start feeling worse, I'm thinking about trying to record Chapter 6 lecture this weekend and just posting it on YouTube and giving you the link, because there's a lot of information that I want to make sure that you get. But I can spend 3 weeks on Chapter 6, and we don't have time to do that. So I may, if I don't start feeling worse, try to record a good chunk of that this weekend and have you listen to it before class and before we get to that, and then we'll just discuss it, and I'll not actually lecture to you, but you can actually listen to it and watch it there. So I'll let you know if that happens so that you will know where to look to find it. But chapter 5 is pretty interesting. Chapter 4 was growing organisms. And so that's interesting, and some students get excited about that, but most students want to know how to kill organisms. And so that's what chapter 5 is about, is killing organisms. And the historical glimpses is pretty interesting. You already know Joseph Lister. How do you know his name? Listerine. So we've got that antiseptic mouthwash that's out there. Why did I call it antiseptic? What does antiseptic mean?
It reduces your chance of sepsis.
Yeah, and what does so antiseptic reduces the chance of sepsis, and it's something that can be used in association with skin to reduce infection possibilities. So Lister was a surgeon, and he recorded a lot of things about what was going on medically back then. And if you read through his annals, it's pretty interesting. Back then it was more dangerous to go in and have just a routine surgery than to not have it. Even having a surgery for something like an appendicitis, you were much more likely to die if you had the surgery than if you didn't. And if you read through his writings, he recorded surgeons going to one table operating on one patient, and then when they finished with that patient, the surgeons would wipe their scalpel on their boot leather, he described, and then move to the next patient. That was the method of sterilization that was used. He'd work on that one, and then they'd wipe the blood and guts off and move to the next one. And it sounds pretty barbaric, but that's the state of medicine at that point. You already know from the first couple days of lecture that there's lots of organisms on the shoe and the foot, and this guy might have had a bacterial infection, and they moved on over to the next person and transferred that. And this was way before the development or even conception of antibiotics. And so lots of people were dying. Basically, the immune system had to win. If your immune system didn't win, then I guess it was survival of the fittest, and then you didn't make it beyond that. And so Lister's methods saved lots of lives. He basically revolutionized surgery and a lot of medical procedures and helped prevent wound sites from getting infected. He knew about Pasteur's research. Pasteur was beginning to describe those microbes that were out there and some of the activities they could do in metabolism, and he started to wonder if they might be involved in the infections that people were seeing. And so he actually introduced using carbolic acid. Some of you had a chemistry class, and you may have played with phenol in chemistry class. Phenol, if you use a concentrated solution, is actually pretty caustic. It'll burn your skin, and so he used very dilute solutions of carbolic acid or phenol to inhibit the growth of organisms, and he found a dilute enough concentration that wouldn't severely inhibit the healing process. And so then that inhibited a lot of growth and prevented a lot of infections of bacteria in those wound sites. And he used that material on a lot of different things to try to prevent those infection processes from happening. As a result, saved a lot of lives. And then he started looking at the surgical instruments and thinking that maybe there was a better way to disinfect or at least get rid of some of the microbes. And so they started using the same solution to clean those. And that made major advances in the effectiveness of the medical practices at that point. It's not very good for repeat business if your patient dies every time you perform a surgery, and so helped a lot. But even in the late 19th century, people were seeing they really didn't understand how infection process happened. And people that were going in for those minor surgeries were having these fatal infections. And physicians didn't even know that their hands could pass disease from one patient to another and at that time. And it wasn't really until the 1940s that this really got better, because at that point we developed antibiotics. The first antibiotic came out in about 1945, and that helped a lot, because then if the patient developed an infection, most of them we could actually hit with penicillin at that point. And so that was a big leap. But today, most hospitals have some pretty strict procedures that are supposed to be followed. And sometimes they get followed, but sometimes the staff gets so busy that they just can't follow them and get everything done that they need to. And so we still have problems today. Surgeries today are obviously performed in a relatively safe environment and procedures are safe, but all the time you hear about a scalpel or a sponge or other things being left in the surgical site when it's being closed up. And so there's all that stuff that happens. And healthcare associated infections are still a major problem. It's a $1 billion problem every year. People getting other infections when they go in to be cared for in a healthcare environment. So Lister made great leaps and bounds in moving things forward, but we still got a big problem in these healthcare associated infection processes. And it's a big problem. It's so big that the insurance companies have started to say, hey, our patient went into your care for this problem. And while they were in your care, they got this second infection that's totally not related. They didn't have that infection, and they picked up the organism that's causing the disease while they were in your care. So we're, as a healthcare insurance agency, not going to pay for that. That's your responsibility as a medical company that caused this disease in our patients. And the hospitals and clinics and other medical companies said, well, wait a minute, that's a major expense. We can't afford to cover that cost either. And so the doctors, nurses, other medical professionals realized, well, it's not always our fault that the patient gets this disease when they're in our care. And so they've started, if a patient comes in for inpatient, they started swabbing noses because one of the top 3 health-associated infections is Staph aureus. And I already told you that about 30% of the U.S. population has Staph aureus already in their nasal passages. And so they've started to swab the nasal passages to, one, see if the patient has Staph aureus in their nasal passages, and if they do, which strain of Staph aureus is there. And they actually save that culture just in case the patient develops a secondary infection. If they do, then they can look at the strain that's causing that infection and look at the original strain, and if they're not the same, then it's probably something they picked up in the hospital. But if it's the same strain that they were already harboring, then they can say, look, your patient, they can say to the insurance company, your patient already had this strain. It's not our fault that the patient transferred it to the surgical site. And so it is your responsibility to deal with that. And so both sides are taking precautions to protect their pocketbooks. Hopefully, in the end, it's protecting the patient as well and preventing some of those healthcare-associated infections. But it still is a huge problem. So we try to control, but you know in a hospital situation there's a ton of sick people, and many of those have immune-compromised systems. Their immune system's not working as well as it should, and many of the others have infectious agents that are causing disease. And when you get those two populations together, there's much more likelihood that you're going to have a problem in that environment. So it is difficult to control. We need to take some special approaches to control in those environments. And so we're going to just get some terminology here for you all. And the first one you already know, it's sterilization. It means that there are no microorganisms, there are no viruses in or on that material. And so the item is called sterile. The next one's called disinfection, and disinfection implies that hopefully all, but at least most of the pathogens are removed from that material, and so that would be organisms that can cause disease. There may still be some viable microbes there, but there shouldn't be many that can cause disease. Yeah?
So if something has a prion on it, is it still considered sterile?
That's a beautiful question, and we don't have a great answer for that, but typically sterile says no microorganisms and no viruses. Since we can't detect prions on surfaces, then it's hard to know whether they're there or not. And so prions usually aren't considered when we use the word sterilization, because right now we don't have a way to know if they're there or not. So. All right, so we can do disinfection, and again, that gets rid of most, hopefully all of the infectious agents that might be there. And those chemicals that we might use are called disinfectants. They can be used on inanimate objects. Sometimes we call them biocides, germicides, or bactericides. If we use them on skin, though, they get a different name. Then they become antiseptics. And that that's the word I was getting to, but then I realized when I asked the question, we hadn't actually talked about it yet. So an antiseptic is a disinfectant that is used on skin, okay? And so some of you have those in your purses or maybe your backpacks. If you have those little bottles of Purell, those are antiseptics. And so because they're a disinfectant that can be used on the skin, and there's lots of different types that are out there. And then if we don't think about our body or surfaces, but think about foods and other things, we can use the word pasteurization. Pasteurization, by definition, uses heat. That's the way Pasteur described it. It uses heat to reduce the number of spoilage organisms. And it should destroy all pathogens. So after pasteurization, there shouldn't be any pathogens left, and the FDA controls that, and that's one of the regulations. There can't be any pathogens in the food that's been pasteurized. There might still be organisms in it. You already knew that, though, because you've all bought a gallon of milk and you drank part of it, and a week or so later it started tasting a little bit tangy in that week's time in the refrigerator, so the organisms that were still there started to grow, divide, produce acid, and make that environment sour. Acid tastes sour, and that's what's going on there. And so not all the organisms are removed, but the pathogens have to be destroyed in that process. By definition, pasteurization uses heat. Clear. Sorry, my box is not cooperating. Uses heat. But recently, you know that some foods you can't heat, right? Like your fresh lettuce salad, you can't really heat that, or it gets pretty wilted pretty quickly. But in the last 6 months or a year, there have been a number of infections and outbreaks, epidemics that have happened from people just eating lettuce that was harvested, or bell peppers in one case, or another case, cantaloupes that had salmonella that got into those materials. And since we don't cook them, the organisms weren't reduced in numbers, and therefore people got sick. So the FDA has actually said, okay, we can do pasteurization in a different way. We can, since those foods don't survive heat, we can actually irradiate them with gamma rays and kill the organisms with gamma rays. And the gamma rays will be absorbed by the DNA of those organisms and the DNA of the material that's being irradiated. The energy will be absorbed, and it'll kill, basically the DNA is broken and other things are oxidized as the gamma rays go through. And it does a good job of killing pathogens, so we can pasteurize in that way. You're going to see more and more foods that are going to be pasteurized that way. We don't usually use the word pasteurization for inanimate objects, but I guess by definition it could be if you use it to just reduce the number of organisms there and get rid of the pathogens that might be there. And then there's decontamination. That's basically taking a surface and reducing the pathogens to a level that's considered safe. Not all of them are going to be gone. That's pasteurization, but at least it's a safe level. And so it's decontaminated. And then there's sanitized. And sanitized, it depends on what you're talking about. There's different government agencies that control different things, like the kitchens in a restaurant, the restrooms in a restaurant, the seating area in a restaurant, all have different levels of sanitation that have to be observed in that. And so there's no one specific level for sanitation, but sanitation means that there's a reduced microbial population there. Still going to be some, but there's a reduced level, and depends on which situation you're talking about. And then there's preservation. We all do that. We put our food into the refrigerator, maybe put it into the freezer to prevent it from spoiling, so we can adjust the conditions where the food's held. And sometimes we just add something that's bacteriostatic. Bacteriostatic means it prevents organisms from being able to grow. It keeps them at a static number, a constant number. And so we've already talked a little bit about that and adding weak acids into foods to prevent their growth. Do that if you didn't. Did any of you go look at cans of food and the ingredients? Maybe I haven't mentioned it to you guys. Do that this weekend. Go to your cupboard, look at your corn, your fruit, your anything else that you have in a can, and look and see what the ingredients are. Usually down toward the end of the ingredients, there's going to be some kind of acid. Usually it says added as a preservative. See what those are and bring them back to class on Monday, okay? And if you don't see that listed there, look and see if there's any other acids in the ingredients. Some of them, they already put vinegar into the food as they're making the food, and vinegar has acetic acid in it. If it's got that in it, then they don't have to add an additional acid to lower the pH there. Those are added to inhibit bacterial growth. Basically, when we put the food into a can, we don't want any organisms that might be in there to be able to grow. Even if they grow a little bit, they might produce toxins like the botulinum toxin, Botox, and then that's a neurotoxin, and that can cause a lot of problems if you eat those foods that are contaminated with that toxin. This part is pretty fun to me. We've got this dad here. He's an awesome dad. He's teaching his son how to cook. It's pretty politically correct. I like that picture. And they're making something with eggs, so hopefully they took the right precautions and maybe clean those eggs a little bit more, make sure that not too many shells are going to go in. If they didn't do that, then hopefully they're going to cook those eggs and not just do the Rocky thing and drink the raw eggs together as they're beefing up their father and son together. So that's a fun one. And then we got this young lady here working at, I don't know if she's at Luby's or where she is, but she's at some place that's making lots of desserts and foods for what appears to be a buffet. And she's got her garb on with her apron, her gloves, and her hair net, and it looks like maybe a mask. Might be a turtleneck, can't tell for sure. What do you think this one is over here?